potentials, then which generate ionic currents, which generate field potentials. Then after this we get this macro potential, which can be found on the scalp, which we measure then later. So, but what are the main influences on this macro potential? First of all, is that there are intrinsic qualities of neurons. Some of them can be generators of specific rhythms, and with their membrane potentials, all that influences this. Then the second important thing to influence this EEG signal is then the interactions between neural networks. So we have a constant changing pattern of this synchronization and desynchronization of let's say, regional brain cells. So this results in amplitude changes in specific frequency bands. This is basically the basics what we get from the EEG signal that we measure on the hair with electrodes. So first of all, then it's important to mention that EEG has great time resolution, milliseconds, but unfortunately worse spatial resolution compared to fMRI, which was mentioned before. And it shows distinct patterns of activity which can be seen in sleep, wake cycle, and also others which can be determined then later with all this analytical approach. So basic brain rhythms are those, and these are also the frequency bands which are mostly used, but I have to warn you that many of the studies are already inconsistent at this point because they define these frequency bands a little bit different. So, but it can always influence the result at the end a lot. So, what would be the functional role of these brain rhythms? Are these brain rhythms just the coincidental finding that correlate with specific brain functions, or are these really something that needs something in a functional way? So, one of the hypotheses of brain rhythms, of course, which is getting more and more weight in the recent years, is that brain rhythms and oscillations have this role in switching neural networks between different functional states in means of activating or inhibiting neural systems, and of course that influences interactions between different regions. So these are the two methods or computer analysis methods for EEG signal analysis. So with these two, we can actually get both of, let's say, with power spectrum, which is defined as a degree or power of representation of specific frequency band in the signal. We can get from that, it is thought to be a functional correlate of different levels of regional cortical activity or inactivity, which, of course, this is a consequence of regional synchronization or desynchronization. So this power spectrum, this already gives us information about activation or deactivation in certain regions. This is quite similar to fMRI in a way. But then with this second, which we will focus on this second signal analysis method, it is called EEG coherence. And EEG coherence is defined as a measure of degree of similarity, or also said phase locking or synchronization of two distant signals for a specific frequency band. So phase locking is that synchronization is a little bit, in a strictly mathematical sense, it's not true, because the phase between two signals can be also different from zero. But synchronizations can be used only if the phase is zero. So we have really phase locking here and similarity of two signals. So different degrees of this long-range synchronization of oscillations between separate cortical regions for specific frequency band are then determined by this EEG coherence. And this is thought to be a measure of functional coupling, which I was telling you before, or binding communication between separate brain centers. So by analyzing this EEG signal with these two methods, we can actually get the view of two different operational systems of the brain. One is concerned with activations, deactivations, or inhibition, and the other is concerned with the interactions, with synchronization of function between two distant areas.